Imagine a world teeming with life, so small it's invisible, yet so powerful it shaped the entire planet. And it continues to drive countless processes, even, right inside you. Today, we're taking a deep dive into the, well, fascinating world of bacteria. You might think you know bacteria, maybe mostly associating them with getting sick. But this deep dive using sources like Bacteria Wikipedia is really going to show you a different picture. Our mission basically is to explore this incredibly complex, diverse, and often beneficial domain of life. I mean, they've been foundational to Earth for billions of years, and they're still an indispensable companion to you. We'll unpack their ancient origins, their astonishing forms, hidden behaviors, and just their profound impact on everything, your health, the whole global ecosystem. And just a quick thing to start the name, bacteria actually comes from ancient Greek. It means staff or cane, apparently, because the first ones they spotted, they were rod-shaped. Yeah. Okay, so let's talk history. Their roots. Bacteria were among the very first life forms on Earth. We're talking about 4 billion years ago. Just think about that for a second. For about 3 billion years, that's like 3/4 of Earth's entire history, bacteria and archaea too, were the dominant forms of life. It's not just a long time, it means they basically set the stage, didn't they? They shaped the chemistry, the biology that allowed everything else, including us, to eventually show up.
Absolutely. And it wasn't just being there. These early bacteria were total game changers. They were instrumental in one of the biggest leaps in evolution.
Yeah.
Eukaryotes. Like our cells, they actually arose from ancient bacteria getting into these endosymbiotic relationships. Basically, one cell engulfed another, but instead of digesting it, they teamed up. That's how we think things like mitochondria, the powerhouses in your cells, and later chloroplasts in plants came to be.
Wow. So, okay, they shaped early life. After billions of years, where are they now? Just how pervasive are they? Give us a sense of the scale.
Oh, they're truly ubiquitous. I mean, everywhere you can possibly imagine, not just the obvious places like air, soil, water,
Yeah.
but also in like acidic hot springs, radioactive waste, even deep inside the Earth's crust. And the numbers are just mind-boggling. They're an estimated 2 times 10 to the power of 30 bacteria on Earth.
10 to the 30. That's a lot of zeros.
It's an enormous number. Their total biomass, like their collective weight, is only exceeded by plants. And think about this, something really relevant to you. The bacteria in the oceans, they produce up to half of the oxygen we breathe.
Half. Wow. So half the air I'm breathing right now is thanks to ocean bacteria?
Pretty much, yeah. It's incredible.
It really is incredible. And you mentioned extreme places. Talk about resilience. These are the extremophiles.
Exactly. They survive temperatures from minus 15 Celsius in Antarctica, way below freezing, up to 121 degrees C in hydrothermal vents. That's boiling. Plus, they can endure extreme radiation one type. Deinococcus radiodurans can take 5 million rads, high salt, even the vacuum of space.
It's just amazing how something so small can be so tough. And speaking of small, let's zoom in. How big are they typically?
Typically, yeah, they are tiny. Bacterial cells are usually about 0.5 to 5.0 micrometers long. That's roughly maybe 1/10 the size of one of your own cells.
Oh, okay, microscopic mostly.
Mostly. But here's something that might surprise you. Some species, you can actually see them with the naked eye.
Really? Without a microscope?
Yeah. Think about Margarita magnifica. It can reach up to 2 centimeters in length.
2 centimeters, that's huge. Like 50 times bigger than other known bacteria.
Exactly. Imagine seeing a single tiny white thread, and that's one bacterium.
Yeah.
It kind of changes your whole picture of them, doesn't it?
It absolutely does. Wild. And it's not just size, right? Their shapes vary a lot, too.
Oh, definitely. They come in all sorts of shapes. Spherical ones called kochi, rod-shaped ones called bacilli.
Like the first ones discovered.
Right. Then you have curved ones, vibrio, spiral ones, spirilla, and tightly coiled ones called spiritetes. And this variety isn't just random. It's determined by their cell wall and cytoskeleton. And it actually affects how they get food, stick to surfaces, move around, even how they escape predators. Shape matters.
And they don't always live alone, do they? You mentioned multicellular structures.
That's right. While lots exist as single cells, many bacteria form complex multicellular groups. Myxobacteria, for instance, can clump together to form these things called fruiting bodies. You can get up to 100,000 cells in one, with different cells doing specialized jobs. It's almost like a tiny, primitive organism.
Like a microscopic colony. And that ties into biofilms, doesn't it?
Exactly. That's a really important concept. Bacteria often stick to surfaces and form these dense mats called biofilms. And they're not just messy piles, they're quite organized. They have channels for nutrients to flow through, almost like little cities. In nature, actually, most bacteria live in biofilms, not floating free.
Okay, so they build communities. What's the relevance for us, for you listening?
Well, it's hugely relevant, especially in medicine. Bacteria inside a biofilm are way harder to kill, sometimes up to 500 times more resistant to antibiotics than individual free-floating bacteria.
500 times.
Yeah, so this makes them a really big problem in chronic infections or like issues with medical implants, catheters, things like that. It explains why some infections just don't seem to clear up easily.
Right, that makes sense. So, okay, they build these tough communities, survive extremes. What's going on inside the cell? How are they built differently from, say, our cells?
Well, a key difference is that, unlike your cells, bacteria typically don't have those large membrane-bound compartments, like a nucleus to hold their DNA or mitochondria. Their genetic material, usually a single circular chromosome, just floats in an area called a nucleoid. And their cell wall is really unique. It's made of something called peptidoglycan. That specific material is actually the target for many antibiotics, like penicillin. That's how those drugs work. They mess up the cell wall construction.
Ah, okay. And that cell wall difference, that's related to the Gram stain test, isn't it?
Precisely. It's a classic test. It separates bacteria into gram-positive. They have a thick peptidoglycan wall that holds the stain, and gram-negative, which have a thinner wall but also an extra outer membrane.
And that difference affects which antibiotics work.
Exactly right. That outer membrane in gram-negatives can act as an extra barrier. Plus, many bacteria have cool external structures too. Flagella, which are like tiny propellers for swimming.
Little motors.
Yeah. Fimbriae, which are like little hairs for sticking to things. And pillili, which are really interesting. They can be used to transfer genes directly to another bacterium.
Gene swapping. We'll come back to that.
Definitely. And sometimes they have a slimy outer layer, a glycocalyx or capsule, that can help protect them from your immune system and also helps them form those biofilms we talked about.
Okay, so lots of tools for survival. But some take it to a whole other level, right? The ultimate survival mode.
Oh, yes. Some gram-positive bacteria, like the ones that cause anthrax, bacillus or tetanus, and botulism, clostridium, can form these incredibly resistant dormant structures. They're called endospores. These things are truly remarkable. They basically shut down, show no metabolism, and can survive almost anything intense. UV light, gamma radiation, boiling heat, freezing, disinfectants, even the vacuum of space.
That sounds almost sci-fi. And they last a long time.
An astonishingly long time. Our sources mention spores found in amber that's like 25 to 40 million years old. Million. And even spores revived from 250 million-year-old salt crystals. They can stay viable just waiting for geological timescales.
250 million years. It's hard to even comprehend that. So what does that extreme resilience mean for us, practically speaking, especially with those diseases you mentioned?
Well, it means those bacteria are incredibly hard to get rid of. You can clean the surface, but if spores are there, they might survive and then reactivate later when conditions are right. That's why anthrax, tetanus, botulism are such persistent threats. The spores just wait.
Makes sense. Okay, moving beyond just survival. Let's talk about what they do. Their engines, metabolism, growth. How diverse are they and how they get energy?
Astonishingly diverse.
Yeah.
Far more diverse than animals or plants. Bacteria can get energy from sunlight like plants do, or from a huge range of chemical compounds, organic and inorganic, even things like methane gas or hydrogen sulfide. This metabolic flexibility makes them absolutely crucial for cycling nutrients on Earth. Things like nitrogen fixation, turning nitrogen gas from the air into fertilizer for plants. Only certain bacteria can do that. And decomposition, breaking down dead stuff, mostly bacteria and fungi, ecosystems would just collapse without them.
And they impact our world in other ways too, right? Both good and bad.
Absolutely. For instance, certain sulfate-reducing bacteria are actually responsible for producing toxic forms of mercury for places like swamps or sediments. That gets into the food chain.
Not good.
Not good. But on the flip side, fermentation by bacteria gives us loads of foods. We enjoy cheese, yogurt, pickles, sauerkraut, soy sauce. Even the ethanol in beer and wine often starts with yeast, but bacteria play roles too.
Right. And how do they reproduce? Mostly just splitting in two?
Yeah, the most common way is binary fission. Basically, the cell grows, duplicates its DNA, and splits into two identical daughter cells. It's asexual reproduction. And under good conditions, some can do this incredibly fast, like doubling their population every 17 minutes.
Every 17 minutes. That's exponential growth on hyperdrive.
It really is. That's why a bacterial infection can take hold so quickly.
But wait, if they're just making identical clones, how do they evolve and adapt so effectively, especially to things like antibiotics?
Ah, that's the crucial bit. They aren't just making clones. They have high mutation rates, for one. But the real key is horizontal gene transfer.
Okay, you mentioned that earlier with the pili. How does that work?
Right. So there are a few ways. Transformation is when they just pick up loose DNA from their environment. Transduction is when a virus accidentally carries bacterial DNA from one cell to another. And conjugation, often using those pili, is when one bacterium directly transfers genetic material to another. It's like bacterial, well, not sex, but gene sharing.
And this isn't just within the same species?
No, that's the amazing part. They can share genes between different species, even quite distantly related ones sometimes. This rapid exchange of genes is a massive factor in evolution, and it's exactly why antibiotic resistance can spread so terrifyingly fast through bacterial populations. A resistance gene can pop up in one place and get shared around.
That explains a lot about the challenges we face there. Okay, so they're metabolically diverse, reproduce fast, swap genes. Do they, like, behave? Do they move with purpose?
Bacteria, absolutely. Many bacteria are modal. They can move, often using those flagella, the little propellers, or some use other methods like twitching or gliding across surfaces. And their movement isn't random. They show behaviors called taxes, moving towards or away from stimuli, like chemotaxis, moving towards chemicals, maybe food, or phototaxis, towards light. Some even respond to magnetic fields, magnetotaxis. They navigate their world.
So they're not just passively drifting. Do they coordinate? You mentioned biofilms. Is there communication involved there?
Yes, very much so. Especially in biofilms, bacteria act like multicellular communities. They communicate using chemical signals. It's a process called quorum sensing.
Quorum sensing, like sensing when enough of them are present.
Exactly. When the population density reaches a certain level, a quorum, the concentration of signaling molecules gets high enough to trigger coordinated group behaviors. This could be releasing digestive enzymes all at once to break down something large, or ramping up toxin production, or even switching on bioluminescence, making the whole colony glow. It makes the group far more effective than just individual cells acting alone.
Wow. So they have social networks, basically. It's clear they're incredibly sophisticated. Now, let's bring it back to us. How do all these abilities translate into their interactions with us and the broader environment?
Right, their roles are indispensable, really. Let's start with the ones living with us commensals. Millions, billions of bacteria live on your skin, in your mouth, your airways, and especially massively in your gut. This is your normal flora or microbiome. Usually they're harmless, just living there. Sometimes they can even be beneficial. But they can cause trouble if they get into the wrong place, right? Like E. coli from your gut causing a urinary tract infection, or bacteria from your mouth getting into the bloodstream and potentially causing heart issues.
Okay, so location matters. And then there are the definite good guys, the mutualists.
Exactly. The relationship is beneficial for both sides. Your gut flora, maybe over 1,000 different species, is a prime example. They are absolutely essential. They help train your immune system. They synthesize vitamins your body can't make, like folic acid, vitamin K, biotondo. They break down complex carbohydrates you can't digest. And here's a big one, vitamin B12. Pretty much all animals need it, but only bacteria and some archaea can actually make it from scratch.
Yeah.
So fundamentally, most animal life depends on these microbes for B12. Without them, we'd have a major deficiency.
That's a fundamental connection, isn't it? We literally can't live without them. But of course, we have to touch on the pathogens, the disease causers.
Yes, unfortunately. While most bacteria are harmless or helpful, some are definitely pathogenic. They cause diseases you've heard of: cholera, syphilis, anthrax, tuberculosis, tetanus, Lyme disease. The list is long. Some are opportunistic pathogens. They usually only cause disease if your immune system is weakened or if they get into a sterile part of your body. And they can cause disease in different ways. Some invade tissues, others produce powerful toxins, like the botulinum toxin, causing botulism, is one of the most potent poisons known.
And fighting these is a constant battle, especially with that resistance issue we discussed.
It's a huge global health challenge, yes. Developing new antibiotics and using the ones we have wisely is critical.
Okay, beyond us directly, bacteria are also huge in technology and industry, aren't they? You mentioned food.
For thousands of years, we've used bacteria, often without even knowing it, to make fermented foods.
Yeah.
Cheese, yogurt, soy sauce, sauerkraut, kimchi. It's all microbial action.
And cleaning up messes, bioremediation.
Yeah, they're amazing bioremediators. Certain bacteria can naturally break down pollutants like oil spills, industrial waste, pesticides. We can harness them to clean up contaminated environments. It's often much greener than chemical methods.
And in farming,
There's Bacillus thuringiensis, usually called BT. It produces a protein toxic only to certain insects. So it's used as a natural, targeted biopesticide, much safer for the environment and other organisms than broad-spectrum chemical pesticides.
and then there's the whole world of biotech.
Absolutely. Bacteria, especially E. coli, are the workhorses of molecular biology and genetic engineering. They're relatively simple, grow fast, and we understand their genetics well. Scientists use them to study genes, figure out what they do, and even engineer them to produce useful proteins. Think about insulin for diabetics.
Hmm.
Most of it today is produced by engineered bacteria or yeast. Same for growth factors, vaccines, enzymes. They're like tiny programmable factories.
It's just incredible the range of impact from these invisible organisms. It makes you wonder, how did we even discover them? For most of human history, we had no idea they existed.
Right. The story starts in 1676. A Dutch draper named Antony van Leeuwenhoek. He wasn't a trained scientist, but he was brilliant at grinding lenses and built these amazing single-lens microscopes, far better than anything else at the time. And he looked at everything pond water, scrapings from his teeth, and he saw these tiny moving things he called animalcules, little animals. He was the first human to see bacteria and other microbes
And nobody else could replicate it for ages.
for over a century. His microscopes were that good, and maybe he kept his methods a bit secret, so the field didn't really take off immediately.
Then the name bacterium came later.
Yeah, in 1828, Christian Gottfried Ehrenberg introduced the term. But the real breakthrough in understanding what they did came later. Louis Pasteur, around 1859, showed that microorganisms were responsible for fermentation and also spoilage. This was huge support for the germ theory of disease, the idea that microbes cause illness.
And Robert Koch solidified that, right?
Exactly. Koch, a German physician, rigorously proved that specific bacteria cause specific diseases, like anthrax and tuberculosis. He developed methods for growing bacteria in pure culture and established Koch's postulates criteria, still used today to link a microbe to a disease. Pasteur and Koch really founded microbiology as a science.
And then a weapon against them, antibiotics.
The first targeted chemical one came in 1910. Paul Ehrlich developed salvarsan to treat syphilis. Then, of course, Alexander Fleming's accidental discovery of penicillin in 1928, though it took until WWII to mass produce it. That really revolutionized medicine.
And finally, a more recent shift in understanding their place in the tree of life.
Yes, a really fundamental one in 1977. Carl Woese was studying ribosomal RNA sequences, basically, core genetic code, and he found that prokaryotes, the simple cells without a nucleus, weren't one group. They were actually two distinct domains of life, as different from each other as they are from us eukaryotes. He named the second group archaea. So now we have the three-domain system: bacteria, archaea, and eukarya. It reshaped our whole understanding of life's evolution.
Wow, what a journey. Okay, so to wrap up, from being Earth's first inhabitants, literally shaping the planet, to being essential partners inside our own bodies, to tools in industry, and sometimes dangerous foes. Bacteria are so much more than just germs.
They really are. Incredibly diverse, incredibly resilient, and absolutely indispensable to life as we know it.
And you now have, hopefully, a much deeper appreciation for these unseen forces constantly at work. But here's a final thought to leave you with, something that really struck me from the sources.
We've only really studied like maybe 2% of all the bacterial species out there. And most of the research, maybe 90%, it focuses on less than 1% of bacteria, mostly the ones that cause disease.
So think about that. What incredible things might the other 98% be doing? What undiscovered roles, what revolutionary technologies or medicines might be waiting in that vast, unexplored microbial world?
It really makes you wonder, doesn't it? The biggest discoveries about life on Earth might still be hiding in plain sight, in the microscopic realm, right under our noses, or maybe even inside them.